Okay. So yes, this is the first meeting of the season of SPAAMTisch. Thank you everyone as usual for joining in. And thank you, Davide, for accepting the invitation and for signing up.
Thank you.
Davide holds a degree in biology and a master from University of Milano in Bicocca, where he studied basically gut microbiomes with a master internship in Copenhagen as well. And during his PhD, then he shifted to ancient metagenomic at the University of Lausanne. And today he will actually present the results of his published work. So thank you, Davide, and the screen is yours.
Okay, thanks a lot, Maria. Let's go, let's start. Okay, so I hope you see my presentation mode. If not, just let me know. Okay, so thanks a lot, Maria, and all the organizing for having me here today. So what I'm going to show you is like an overview of the recent work we published with the title of Towards Predicting the Geographical Origin of Ancient Samples with Metagenomic Data. So just to give somehow some background, when I started my PhD working on ancient metagenomic, I soon realized that one of the biggest challenges faced by the field is dealing with samples, and specifically dealing with problematic samples, such as samples that lack some annotation or samples that have been at some point mislabeled. So it's very common for people in the field to work with a museum collection, and these kind of issues continually arise. So when I say where in the world, here I mean facing the problem of dealing with samples of unknown or sometimes wrong origin. I say wrong meaning like mislabeled samples that are for some reason assigned to a location which is not the one they belong to. Now, this translates to a series of problematics. So here I'm just touching on a couple. Of course, this translates to problem in data analysis, but also, for example, in repatriation practices. So just to have a couple of simple examples here, let's suppose we have an individual that has been found according to the museum label from location B, but shows genetic ancestry similar to people from location A. So the obvious interpretation of these would be somehow the movement of people from location A to location B. However, if this was the result of a mislabeling, it's clear that the outcome is somehow a false mobility pattern, or at least biased mobility patterns due to the mislabeling. Also, this can translate problem when we are trying to repatriate samples from a museum to the community that to which the samples belong to, and of course, if the sample was mislabeled, this could lead to the repatriation to the wrong ethnic group. So there are, of course, approaches that can deal with these issues. Of course, you can study the human, or more in general, the host DNA to study the genetic ancestry of the individual. And this can, for example, inform us about who is the appropriate ethnic group to which a given samples belong to. But of course, the retrieval of human DNA is not always possible, and when it comes to like studying mobility, for example, having the human DNA is not really helping us to rule out movements of people because it tells us nothing about the place where the actual individual died. Isotope analysis has been used to study, for example, where an individual lived. So, for example, the use of a strontium isotope ratio has been used to identify individuals that are somehow that moved from one region to another place during their lifetime and showed somehow non-local isotope ratios. However, of course, there are limitations also for isotope analysis, such as the reliance on reference panels that are not always available. But more specifically, none of these two approaches can specifically address the question of where an individual died, meaning where the remains come from. And here is where metagenomics can come to rescue. So people from this community knows what a metagenome is, and knows very well that when we sequence DNA from an ancient sample, what we get is a mixture of host DNA, usually in smaller proportion, and a lot of microbial DNA, which comes from the environment, at least most of it. And I think I don't think I will need to put much effort to convince you about this, but just to show you, if we look at the composition of ancient samples, so here are a few data sets that I've analyzed in this study. We will come back to those later, but here I just want you to focus on the composition. And you can see that if you look at the phylum level, we see that there are some specific phyla that tends to be highly abundant in relative abundance, such as the Pseudomonadota, previously known as Proteobacteria, or the Actinomycetota or Actinobacteria. And at the genus level, you can see there are some specific genera, mostly of soil-dwelling or ubiquitous bacteria that are found at high relative abundances. And if we just try to plot based on a compositional pattern, the samples from different sources. So here in this case, some bone and teeth samples in blue and purple respectively, together with samples from other metagenomic sources, such as soil or host-associated environments, such as oral cavity, the skin or the gut. You can see that samples from like ancient samples tend to cluster with soil metagenomes. Of course, here you see in the middle, there are some teeth samples specifically that tends to lean more towards the right. And this is probably due to the presence of also some oral taxa in these kind of samples. However, I think we have seen this already in different publications before. Metagenomic data from ancient samples are dominated by DNA from soil-derived microorganisms. So can we make use of this? Specifically, we ask ourselves, can metagenomic data inform us about the geographical origin of a given sample? So let's take this example. Let's suppose we have samples from two different locations. So the hypothesis of the study was individuals from the same geographical location will share more similarity in their metagenome. Or in other words, we wanted to ask, can we distinguish samples from different geographic location based on their metagenome? So here, in this study, we tested the approach on two different binary data sets. So one data set included samples that we were analyzing in the lab. So it's the one you see on the left. It included samples from Polynesia, 15 individuals, and from Brazil, 24 individuals. Then I also tried to recover other data set that could be used to test this idea. Specifically, I found a data set that was where samples were generated through to the same laboratory procedures at the same time. So this data set allowed me to somehow control or bypass batch effects and somehow assess my method on an ideal scenario, which, as you probably already are thinking, it's usually not the case. But we will come back to this later on. Here, just wanted to go on through a few reads preprocessing steps. So before any kind of analysis, we removed all the human DNA from the data by mapping to the human reference genome. We also removed known sequencing contaminants that are used during sequencing step, such as the phage X. We removed low complexity region, and we also removed all the fragments longer than 80 base pairs. So this was somehow a strict filter that allows us at least to control for modern contaminants, expecting like modern DNA sequences to present longer fragments. So there's many ways that we can use to compute metagenomic similarities. Classical approaches usually rely on compositional, on the comparison of composition of different data sets. These require a classification step, and which is to a certain extent more time and resource intensive, and also usually leads to the loss of part of the data because for some microorganisms we cannot achieve a good classification. So what we relied on for this study was a k-mer-based approach, which is faster and it takes advantage of all the sequenced reads. So just a schematic explanation of how this works. So we compute k-mer spectra. So here is a toy example of a sequence for which we compute formers, a spectrum of formers. So you basically generate sequences of 4 letters shifted by 1 base pair. And this is what is called a spectrum. You generate these for all the reads for within a given samples and for all the samples. And then you can compare these spectra and identify and compute similarities score between samples. So this basically generates a distance matrix that then can be used to perform an MDS. So we generated the distance metric and performed the MDS for the 2 datasets I've shown before. And here is what we observed. So we can see that the k-mer based similarities reveal geographic dependent samples clustering patterns. So here on the left, you see dimension on the upper plot, dimension 1 and 2, and below dimension 1 and 3 for the Brazil and Polynesia dataset. And you can see that already visually, these samples tend to cluster apart. And similar observation can be made also for the Denmark-England dataset. So that was nice, but we wanted to try to assess this in a more quantitative way. So we asked ourselves, can we train a model to predict a sample geographic origin based on metagenomic similarities? So here is like a brief explanation of the methodology behind. What we use is a logistic regression model with the lasso regularization to control for overfitting. The aim is to perform a binary classification. So we have this binary data set, Polynesia, Brazil, and Denmark, England, and we want to try to differentiate samples coming from the 2 locations. The model training was done by giving in input the MDS dimensions. And the lasso regularization was applied to perform automatic selection of informative dimension. And to test the model, what we did, we rely on a jackknifing approach since since the data set are quite small, we cannot somehow divide samples in training and testing data set. We decided to just rely on a jackknifing approach. So here, every time before training a model, we remove 1 sample, and then we try to predict the samples that was not used in the training. And this is done for all the sample in the dataset. And the output of the classification is basically a probability of a given sample to come from one of the 2 locations. Now, here is a ROC curve that summarizes the performances of the model when trained in the 2 different datasets. So in green, you see the Brazilian Polynesia dataset performances, and in blue, the one on the Denmark-England dataset. Here you see I've split these in 2 analyses. In one analysis, I included only samples with more than 1 million reads, and which is the blue dashed line. And in the other case, I include all the samples that I had available. As you can see, where we include all the samples, also the one with less reads, the performance tends to be the model seems to perform slightly worse, which is expected because in the end the approach relies on the availability of data, and the less data we have, the less likely we will be able to correctly estimate metagenomic similarities. But the main take home of this slide is that the model performs very well. So it seems that we can predict the origin of these test samples. So this is somehow the first take home of the paper. So metagenomic data contain information that can be used to trace the geographic origin of ancient individual sample. We also run an analysis to assess the effect of the k-mer length on the performances of the model. And we tested different k-mer lengths on the different data set analyzed here. And the main observation is that the choice of k-mer length is not obvious and can be dataset dependent. But overall, it seems that short and long k-mers should be avoided. And the reason is kind of obvious because short k-mers will tend to have less, to yield less specific information, while longer k-mers will tend to reduce the overall computed similarities. So here is just on the right, you see a series of plots of the decrease in the proportion of correct classification of the model at increasing probability threshold. So basically it's the confidence of the classifier. So the more to the right you go, the more confident is the classifier, and you can see that the yellow line, which is the shortest k-mer, 11 base pair, in some cases perform pretty badly compared to the others. But the same can also be observed in some cases for the dark red one, which is the longest k-mer. So overall, there's not a universal best k-mer length. It should be tailored probably to the data set, but very long k-mer or very short k-mer should be avoided. Now, okay, that was nice to observe, but of course, prediction can be affected by both the variable of interest, meaning geography, but also technical biases, such as laboratory procedures or the sample type, and it's quite natural that these two variables, meaning the variable of interest and the technical biases, will tend to correlate. And this will complicate the analysis because we will not be able to distinguish the effect of the biases. So in the first data set that I've analyzed, we had a couple of potential biases that were partially but not perfectly correlated with the variable of interest. So specifically, the extraction protocol, which it seems to highly affect the composition of the retrieved metagenome. And so it's a very important bias to control, and the sample type. So here you see, again, the MDS as before, but here I'm also encoding the information about the extraction protocol on the left and the sample type on the right. Just going to the performance of the model, what we did was training the model to predict these other variables, meaning the biases. And here is a summary. So you see again, for the Brazil Polynesian dataset alone, because it's the only one that where we have variation in the protocols. You see in green, the geographic location prediction performances, and in the purple, the extraction protocol prediction performances, and in orange, the sample type. As you can see, geography is the variable that is predicted better, but also the other two variables tends to be predicted pretty well. Now, this can be a result of these variables being partially correlated with geography, but overall, these highlight the high risk of technical procedure bias in the model. So what do we do about it? Can we control batch effects? So, and here we comes to the idea we had. So if we look at the microbial composition, there are some specific taxa that are, some specific genera that are present in high relative abundance proportions. So, and what we thought is if we just focus on data that comes from a given bacterial genus, then we mostly bypass the effect of different extraction protocols, as we expect extraction protocol to differentially extract DNA from species that shows different biological features. But within a single genera, we expect species to be kind of mostly similar between each other. So we do not expect from a theoretical perspective to have big changes in their DNA retrieval. As the protocol change. Also, looking at specific genera can allow us to exclude genera that are known to be present in the laboratory kit reagents. So known laboratory contaminants. We already have a good knowledge of which genera tends to contaminate laboratory reagents, such as Pseudomonas. So we can try to exclude these known contaminants. So the way we decided to go was, okay, we have a lot of streptomyces in this ancient metagenome. It seems to be always present and seems to be present very often at high relative abundance. So we decided, can we have a look just at the streptomyces read and use those to perform a geographic origin prediction? Streptomyces are a well-known soil-dwelling microorganism, and we know it's not one of the classical laboratory reagents contaminants. So it sounded like a good candidate. So we repeated the same analysis as before, but using only reads that were classified as streptomyces. Focusing only on samples with at least 100,000 streptomyces reads. So we discarded a few samples that didn't meet this condition. But of course, streptomyces reads are just a very tiny proportion of whole metagenomic data. So we actually had two questions we wanted to answer. First one, will streptomyces data alone contain enough information to differentiate between geographical location? And secondly, will the use of streptomyces data mitigate the effect of technical biases, which was somehow our hypothesis, at least in comparison to whole metagenome? So this is the answer to the first question, as you can see. Well, here I'm comparing the performances of the model when trained on whole metagenome with the continuous line and when trained on streptomyces data only. So here you can see that the performances of the model are actually increased. The model is performing better when using only streptomyces data, which was not just good, but even better. And if we look at the other two variables, meaning the biases, we can see that the performances for the extraction protocol prediction and the sample type prediction are reduced compared to whole metagenome. So supporting our initial hypothesis. So here are just four main take-home of the study. So first is a reiteration of what has been already observed in literature. Metagenomic data from ancient samples are dominated by DNA from soil-derived microorganisms. These data contain information that can be used to trace the geographic origin of ancient individual samples. We show that K-MER-based approach are a good option to estimate metagenome similarities. And lastly, and most importantly, we observed that streptomyces data can trace a sample geographic origin while mitigating for batch effects. So this holds it seems like a promising approach when it comes to meta-analysis that wants to integrate data set from different studies. So there are, of course, some limitation that relates to both the study and also to the approach that I presented today. So regarding the study, to a certain extent, the approach was tested only on a limited number of data sets. So it just counts for 90 samples from 4 different locations. So it still remains to be answered to which extent are these observations generalizable to other locations. Also, the absence of soil samples from the site somehow didn't allow us to compare directly the metagenomes of the bones to the one of the soil from the location where the sample came from. And regarding the approach, there are some limitations which are to a certain extent common to other approaches such as isotopes. What I presented rely on the availability of reference panel to conduct any type of analysis. And it's also tailored just for binary classification problem at the moment. So it's unclear how we can somehow extend this to multinomial classification problem. So meaning having more than 2 locations in the prediction step. There's also some open question that I want to highlight. So first of all, how does the proposed approach behave at different geographical scale? This remains to be explored. How environmental factors such as climate or soil type can affect and potentially bias this type of analysis. For example, you can imagine similar soil type more distant geographic location could potentially harbor more similar metagenomes that will translate to more similar metagenome within the bones that we sequence. And of the big question, like the big open question, can we develop more powerful approaches that will allow sample geographic prediction, taking advantage of all the growing number of available metagenomic data set? So the big challenge there, as I have highlighted in this presentation, will be to control for the risk of batch effects introduced by different laboratory procedures, different contamination, etc. So I would like to thank you all for listening, and thanks all the people that contributed to the project, and also the people in my lab. And if you want to read more, as Maria said, you can check out the publication. It's online on Scientific Reports. And if you have any question, I'm happy to answer.
Any question?
I have a question. No. Raise hand, before.
Go, Cami.
Okay. Hey, can you hear me well?
Yes.
Cool. Thank you for the talk. Really nice. And I was also like in the meantime checking a bit the paper. So I had two questions. One, I think you digged a bit at the end of your talk. I just wanted to know how useful the classification method would be if the samples came from a closer region, let's say two samples from, I don't know, Argentina, one from the north, one from the south. If you kind of had the opportunity to test that out, or how could you think this like situation could be assessed? And then another question, if I understood correctly, you performed a PCA and then you use those principal components as input to your logistic regression, right? So was that previous to the separation of the data, the splitting of the data into training and test set, or was that after splitting the data? Mm-hmm.
Okay, so so regarding, well, the first question, like yeah, it's as I mentioned, like we don't know yet how this will behave at different geographical scale. It's something that remains totally to be explored. Of course, I think probably my expectation would be that the resolution will depend on the scale. To a certain extent, if we look at a single site, so samples coming from a specific site, if you want to check a samples come from that very site, it might be easier because to a certain extent, I think we can more, the metagenomic similarities will be much higher. When it comes to like more distant geographical scale, there is also where it becomes less clear. For sure, the metagenomic similarities will decrease, and also other factors such as soil condition might start to play a role and bias the results. So, so this I don't have an answer. I only have question in that sense. And for what concerns the second answer, so yeah, the way it's done. So every time a sample is removed from the training data set, we recompute the MDS, and we use those as input for the model training, and then the sample is somehow projected, and we see like and based on the position of the projected sample, we infer like the model will predict if it comes from one or the other location.
Thanks.
Thank you.
Oh, I have a question actually.
Yeah, good morning.
You mentioned that you removed the human read in the beginning, right?
Yeah.
So, but because in my experience, you did that by mapping to the reference genome. And can I ask which kind of parameters did you use? Because sometimes that kind of restrictive parameters leave some low quality human reads in there. And so I always ask myself, how much can this pull the analysis towards one geographic location rather than another if you are using, you know, associated metagenome.
Yeah, so that's a very good point, of course, because if human data are left in, you might expect that somehow they will play a role in this similarity estimation, and if more similar humans are in there, they will probably inflate their similarity score. So the way we did it is we used an approach. I think I was relying on BWA MEM for mapping to the human genome, so that it's not very stringent, so you actually get to map more stuff than in many other cases. And as a sanity check, actually we checked for some we did some testing. I've not gone into it, but we check if individuals that that showed similarities in their human ancestry, but not in their origin, were somehow they tended to be attracted together or not, and that was not the case. So we expect that probably human DNA is not playing a major bias, but it's something for sure that it has to be kept in mind in this sense, because if human data are there, they can play a role in the similarities estimations.
Thanks.
Are you asking because the not sure if they want to ask because the audio is off? No, they're just talking. Okay.
Any other question, guys? If not, I have others.
Okay.
But please, if somebody, asks before me, bye, Betsy.
Bye, Betsy.
If somebody asks before me, please ask away. Anyone? No. So it's interesting the choice you made about the phyla or like that you chose family, the Streptomyces. You said you choose this because you are sure is not a contaminant, or is because it's host associated and it's also in the soil, like kind of.
Yeah, so the reason were like there was more than one reason. First of all, so there's different studies that have looked into laboratory agents contaminants, and there is a list, I think, somewhere that reports this. So then you know, like if you've been dealing with this, you know, like Pseudomonas is a very known well contaminant. It's also like the second most abundant genus in my data set. But of course, given this aspect, we decided to not use that one use those data to estimate the origin. But the Streptomyces is not reported as a contaminant. It's well known to be a soil-dwelling microorganism. So we somehow were confident enough that we could use this genus as an input for the model prediction.
Okay, but my question is, what if, because you are using, I mean, soil bacteria, say, to trace the origin, no? But what if a soil bacteria is stuck enough to the surface of the bone to be good enough, and like you can trace the ancient signal, and is a good, you know, kind of, can that be a good way of tracking as well, even if it's not, I mean, it's considered a contaminant, but is actually proper of that soil location? Because, you know, I think, I'm not sure, but maybe in forensic science, people use a lot the soil bacteria to define like the soil of that specific place, or
Yeah. I mean, that's to a certain extent is my expectation. So I expect like probably like different location, different area will yield different streptomyces composition. So it's not probably just one, it's probably a mix of species that lives in a given soil. And this DNA somehow ends up in our samples. Can, you know, the exact composition of these exogenous components can be partially microorganisms that take advantage of the bones, so they colonize actively the bones, but also can be like more general species that lives in the surroundings and whose DNA leach into the bone. So it can be more or less active process. But then, yeah, you as you were saying, like you expect different location to yield different microorganisms. So then if you compare these microorganisms, that can be done in different ways. So here the way we do it was like using k-mers, which is kind of a very simple approach. So you just take the reads, it just creates these k-mers, and you check how many are shared. So then in this way, if you look at a specific clade, you expect that the expectation would be like neighboring location or the same site will have same microorganisms, so they will end up in the bones. Different location will yield different, and then you can somehow differentiate them. Not sure if this answered the question.
Yes, no, it does, yes. But did you have to build a K-mer matrix or like some reference panel to do this, or not? What was your reference?
So here the reference I was using was the data set I was the data set I had, right? So I compute basically the spectrum for all the samples in my data set, and then I compared in a pairwise fashion each samples to each other in the data set, and this way I generate the distance matrix. There's I used SORMesh as a tool to do that. It's not the only one available, but yeah, that's the one I used, and it's pretty straightforward to do. It's quite easy to use.
So say if someone wants to start from zero with this method, because I guess it's really quite tailored to a specific case, no? Does like
this, yeah, the one I presented. Yeah, I mean, then I think it has somehow to be tailored to the data set you're looking at, for sure. And you have to be mindful of all the potential biases that are coming into place. So if the data you're parsing is most likely coming from different batches, maybe they use different extraction protocols. This is something you have to be aware of, because it can play a role. So the hope is that using a specific genera such as the streptomyces will help us to bypass some of these biases, such as contamination or extraction protocol differences. And the good thing of streptomyces is that it really seems to be present in high relative abundances in almost all the samples I parsed. So it could be a very flexible candidates also for other dataset if this observation is repeated elsewhere as well.
Go, Cami.
Yeah, I had one more question. Did you test out like scalability? It has to do, I think, with what Maria was aiming at. So maybe running the method on a larger data set, so far more samples, maybe a chunk of the ones in the ancient directory, ancient metagen directory.
No, that I haven't tried yet. No, I think although that if this method has to be applied for in a multinomial context, so I mean we want to have samples from more than 2 locations, that will probably require more sophisticated approaches. So here it was a simple logistic regression. We also tested a random forest approach. They were comparable in their performances, but it's possible that the more data set you want to integrate, that will probably require more sophisticated approach, and also depending on the approach you want to use, it will require a bit more effort to control for the various batch effects and biases. Yeah, but that's somehow the big open question.
Anyone else have questions? Mhm. In the meantime, what kind of bones did you use? Did you know? Only like
So
long
in this
bones,
case, no, in this case it's mostly petrous bones.
petrous bones. Okay, great. Yeah.
Or like when it's a bone sample, when it's a tooth, it's a tooth. It's the dentin. In any case, it's
Yeah, like
so,
the part of the
yeah.
yeah. Yeah, and nobody is asking, so I can keep going for that.
Can keep going.
Because you said as well that it was coming from how many of your samples were coming from museum collections, for example?
Yeah, so this case, I think for the Brazilian Polynesian data set, they both came from a museum collection.
Yeah, and could you see in those ones like some sort of because I felt well, I feel like when they have been, depending how long they have been in the museum, there must be some kind of metagenomic trace of manipulation as well, like skin or
yeah. So that's
taken into account, like
the storage contamination aspect is something that we looked into just briefly. We had the opportunity to look for this effect on few samples because there was published data of samples from the same museum as the one from Polynesian that we had. So same museum, but they were from South America. So arguably more close to the other locations, so Brazil. So to one extent, if the contamination from the museum was present, we would probably observe these samples be classified as Polynesian, let's say, if the contamination was somehow overwhelming. But that's not what we see. So we tried to test with these few samples, and that was not the case. So, but of course, like this is just a small case, few samples. It's entirely possible that in other cases, the contamination during the storage might play a role. And again, here, like one approach to avoid contamination is to somehow focus on bacteria that we know do not contaminate specific environments. So I think we don't have enough knowledge actually about what kind of contaminants can be present in a museum. So that's the only shortcoming in that sense. But yeah, so that's certainly something that can potentially play a role as a bias. Yeah.
So I mean, sometimes they are just chucked in a drawer with so many other bones, probably with soil from other places. So it can be really messy to control.
Yeah, that's for sure.
Well, it's really interesting. I
Yeah,
love
but
it.
it remains to, like, I guess, like, depends on how the samples is pre-processed also. Maybe if some of the contaminants more like environmental contaminants from the museum that remains maybe most on the surface, then maybe you can get rid of them, and then you just have those that were somehow that infiltrated the bone. But still, it's something that remains to be explored.
Yeah, I think it's good the choice of Petrus bones actually, because it's really like kind of, if you are talking about the pars petrosa, so the really inside part of the bone, then I think it's a good choice because it's protected really. So what enters there is really like soil.
Yeah, I mean, to ultimately assess all these aspects, you really need to somehow set up a study with a framework that would allow you to control for these contaminants. In my case, I relied on previously published data, so you can control up to a certain extent these aspects.
Ciao Davide.
Ciao Giulia.
I have a question. I know, like, for example, there are different ways to source, for example, source tracker that we use after taxonomical classification. And I'm speaking right now, if we talk about like sediment samples, for example, where we can get some sources and see if it's like river sediments, I don't know, clayish, or yeah, different sources based on the environment. But do you think this approach, like this K-MER approach, can be also used to track the origin from bacteria, from sediments?
Yeah, and I mean, yes, good. It's a good point. It's a good point, and I would say it's possible, because in the end, like, I'm not entirely sure now. I can't remember how Sort Trackers does the tracking itself. If maybe it's actually using a K-MER based approach itself. I'm not sure.
I think it counts
it just because it's a classification based, right?
Yeah.
Actually, okay, yeah. So it's just counting. Okay, now I remember. But yeah. That's totally possible. I think you could do that. Yeah.
In that case, you need like a reference.
You probably need a reference from soil, reference from other the other potential sources. I actually run Source Tracker on these on the samples I had, and
yeah. I'm reading the chat. Sorry.
Yeah, and you know, the soil is a very variable environment, so most of the taxa remain somehow unmodeled. Like you cannot trace the source. That my intuition is that it's probably environmental soil dwelling microorganisms that cannot be modeled with the soil samples that I used as a source in my modeling. So then it comes a bit to how representative the samples for a given environment, the samples you have for a given environment are, which I guess I would argue for human associated microbiome is we probably have good knowledge of them. You can probably model them very well, but for the soil, I think it's normal that most of the stuff still remain unknown.
Yeah. Thank you.
Thank you.
So I think if nobody has questions, I also finished my set.
Okay.
So,
all right.
I mean, thanks, Davide. Thanks a lot. This was a great talk, and I will stop the recording now.